Hi everyone, I hope you are doing well. In continuation to our discussion which we started in our last class about amino acids, we are going to discuss about the amphoteric nature of amino acids in this class. Then we will see the titration curve of amino acids. Then we will study the titration of glycine in detail, and then we will take few important points from the titration of the glycine and how those can be applied to other amino acids. Okay, so this is going to be a very interesting session, so stay here and just pay attention to the slides. Let's start the class. So if we see, amino acids have the amphoteric nature, so they can act as a weak acids and bases, and that happens due to the amino and carboxylic, and in some amino acids the ionizable R groups. Now you understand that we have this R group as a side chain. So in all the amino acids, carboxylic and amine groups are ionizable moieties, but in few amino acids, R is also ionizable. Okay, and therefore they can act as a weak acids and bases. Now at the physiological pH, which is around 7.4, the acidic proton of the carboxylic acid, so this is the acidic proton, it protonates the amine. All right, so when the proton is lost from the carboxylic group, that becomes a negatively charged, but the positively charged, it goes to the amine, it gives it a positive charge. Therefore now the amino acid has one negative charge and one positive charge, and that is happening when R is neutral. It has no ionizable group. So that makes the molecule a dipolar ion because it has two poles, one is the positive, another is the negative, and such molecules of opposite polarity are called as zwitterions or dipolar ions. Okay, now substances having this dual acid base nature, they are called as amphoteric substances and they are called ampholites. So whether you use the term ampholites or amphoteric, they are the substances with having this dual nature substance. Okay, the dual nature moieties. Now at other pH values, we talked about the biological pH, which is 7.4, where we saw the dipolar ion nature. But other than that particular pH, the molecules become singly ionized. Either they will become positively ionized or they will become a negatively charged group. So they will favor either the cationic or the anionic forms. So here you can see this is where the zwitterions was seen here, one negative and one positive charge. So the net charge was 0. But if you forget this particular pH and you talk about lower pH or higher pH, here you will see that the moiety is either the negatively charged or a positively charged, but not both the charges present like in case of this particular pH. Okay, so all amino acids, they lose protons with increasing pH. So if you see here, we can see here it has the proton, the carboxylic group has the proton at the low pH. Okay, now when you are increasing the pH, so pH here, this is the pH scale, and you are increasing from 2, 3, 4, 5, 6, then it has lost the proton, and then you can see here the proton is lost here. All right, and then further when we move to higher pH in the basic range, then this amine group also loses its proton. So that's why all the amino acids, they lose protons when we increase the pH. So this particular graph, it shows that at a particular midpoint there is a pH where one negative and one positive charge will be there, making the moiety electrically neutral or zero, and such ions are called as zwitterions. But as we move on either side, then we will have either the cationic form or the anionic form. Okay, all right, now let us talk about the titration. And I'm sure, in chemistry, you must have heard this term and you must have done by yourself. So amino acids have the characteristic titration curve. So that means you take any amino acid out of the 20 common amino acids, each single amino acid will have its own titration curve, and that you can easily characterize. So, what is a titration curve for those who just forgot? A titration curve is a graph which shows how pH changes as a titrant, which is an acid or base, is gradually added to a solution. All right. So basically it is a graph which shows the change in the pH when we add a acid or base, which we call it as a titrant, to the solution. Now, if we specifically talk about the glycine titration, which is the point of our discussion for today's class, you take a burette, all right, and you fill the burette with the NaOH solution of the known concentration. So sodium hydroxide with the known concentration, you will fill this burette here, okay, and then you will place the flask with the known volume of glycine solution on a magnetic stirrer. So here this is the flask with the glycine solution, and you know what is the volume, okay, and then you put it on the magnetic stirrer. Why magnetic stirrer? Because magnetic stirrer will uniformly mix the solution when we will add the NaOH, okay. Now we can dip a pH meter, or we can use a pH indicator in the flask to know when the pH will change. It is better if we have the pH meter because it will give us the real time change in the pH. But if you have the pH indicator, which is the color indicator, that will also work, okay. And then to start the titration, we will start adding NaOH slowly, drop by drop, okay. We will put one drop, we will just mix it, and we will see the reading. Then we will see another drop, drop till it starts changing the color. All the pH will start changing. Clear? This is how we do the titration. This was the methodology of doing the titration, and we keep noting whatever changes we are seeing in a notebook. Now we will continuously record the pH after each addition. All right, so every time, and because it is a burette, so you will see how much NaOH has been added here, okay. Where you filled and how much has been used. So based on the reading on the burette, how much NaOH has been added. And then you can also, because you have the pH meter or the pH indicator, you will also have the pH reading of each addition, after each addition, okay, right. Now, if we see the titration curves of glycine, so this is the glycine titration curve. We have two distinct stages. Here you can see two blue boxes. One box is here, one box is here. So these are the two stages, and they show two deprotonations. So deprotonation is, we use the term when we are losing the proton. So that means we are losing two protons here in the titration of the glycine, and that's why we see these two distinct stages here, okay. Now each stage, it resembles in the shape of the titration curve of a monoprotic acid. Now monoprotic acids are those acids which will lose a single proton. So one stage where we are losing the proton, it will resemble just like a monoprotic acid titration curve. For example, here it is the acetic acid. So we have the two stages, and both the stages, they resemble like the shape of the titration curve which we will get for a monoprotic acid. All right, now at a very low pH, so here is the pH, you can see here the pH is 13 here, the pH is 0 here. So we are talking about this particular part when the pH is low. So at the very low pH, the predominant ionic form is fully protonated. All right, so here what you will see for the glycine is the fully protonated form. So carboxylic group also has a proton, which is H, and amine also has a proton which has a positive charge. So we call it as a fully protonated species, and this is the form which you will find at the low pH, okay. Now at first stage, here in the first box, there is a loss of proton from the carboxylic group. So you will see this H, which you can see here, it will be lost. So here you will see the loss of the carboxylic, the proton from the carboxylic group. And the midpoint of the first stage, which is this is the midpoint where my red dot is right now. This is the midpoint. At this stage, it has the equimolar concentration of the proton donor, which is NH3, and COOH, which is the fully protonated form, and proton acceptor, where you will have a lost species here. So equimolar concentrations are there. Okay, here the pH will be equal to the pKa of the protonated group, which is being titrated. Now, what is the protonated group being titrated? The one which can donate proton, and in this particular case it is carboxylic group at the low pH. Understood? So that's how this is the first stage is defined. Now, for those who don't know what pKa is, so pKa is the measure that how easily a group donates a proton, and the scale is logarithmic like the pH scale. Okay, now when we see low pKa, that means it has a high tendency to give the proton, so it is a stronger acid. All right, but when we see a high pKa, it has a lower tendency to give the protons, that means it has a weaker acid. And that's how you see how the relationship of pKa comes here. Now, for glycine, the pH at this midpoint is 2.34. So this is determined 2.34 at this particular pH, or this is the pKa of the carboxylic group. Okay, so we can see here in the graph, we see here it is pK1, which correspond to the pKa of the COOH group. I hope you understood this first stage of the titration. All right, now next important point in the titration when we keep adding NaOH is 5.97. Okay, now this 5.97 is the point where the removal of the first proton is complete, and where it was removed from the carboxylic group, and the removal of the second has just begun. All right, because you can see two stages, two deprotonation stages here. So first proton is removed here, and that is the preparation for the removal of the second proton, or this has started here. All right, now you know the glycine as a zwitterion, here the net charge is 0. All right, and this particular pi, which is the midpoint, you will see the net charge is 0. So here you can see here, it has a 1 negative charge, it has a 1 positive charge, it becomes a dipolar ion, and that's why the net charge is 0 here. All right, now at very high pH, now we are talking about this stage, the high pH. Okay, the predominant ionic form is fully deprotonated. So here this is completely protonated ionic form, where you can see there is no positive charge. So that means the extra proton which was giving a positive charge here, it is also gone, and this was already gone here. So now both the protons are lost, so we call it as a deprotonated form. So this is the fully deprotonated form here, and there was a loss of proton from the NH3 in the second stage. So we already see that how this NH3 has lost its proton, and it has become the NH2 here group. Okay, now the pKa of the midpoint of this stage, it was determined to be 9.6. so here it is 9.6, and we see that complete titration at the pH 12. So somewhere around pH 12 the whole titration is complete for glycine. Okay, now at pH 5.97, if we revisit this particular pH again, this glycine is dipolar because you have equal positive and negative charge. So one charge is negative, one charge is positive, making it as a zwitterion. Now this characteristic pH is called isoelectric point or isoelectric pH. All right, remember this midpoint is known as pi, which is the isoelectric point or isoelectric pH, where the net charge is 0. Okay, now since glycine has no ionizable R group, you know that it has no ionizable R group, it has only H. simplest one. So the isoelectric point is the arithmetic mean of the two pKa values. So we got one pKa value as 2.34 and another pKa value is 9.6. So if we take the average, that comes out to be 5.97. So this is isoelectric point, which is pKa1, pKa2, and then we are dividing it by 2 to get the average, and we get the average of 5.97, and that is true here for glycine. Why? Because glycine has no ionizable R group. All right. Now, glycine has net negative charge at any pH above its pi. So the pi is 5.97. So if you go up 6, 7, 8, 9, 10, then the glycine will have a net negative charge. Remember that. Why? Because you have predominant species as a negative species here. And when you go below the pi, which is 5.97, below is like 4, 3, 2, 1, then it will have a positive charge. But when we see here, when we see the charge, so the net positive charge below pi, it will move towards the cathode, because then it has a positive charge. So the farther the pH of the glycine solution is from its pi, the greater the net electric charge. So when we move farther from this on either direction, either up or down, then we have the greater net electric charge. Why it happens? Because more that particular species which is contributing to the charge is accumulating. Okay, and that's why we have a greater net electric charge. Now the pi of the polypeptide, now we are talking about the polypeptide, we are not talking about a single amino acid. So remember, the pi or the isoelectric point of the polypeptide is not easily predicted. Just in case of amino acids, we can easily predict the pi because we can titrate that. But in case of pi, you cannot predict the pi. Why? Because there is no free alpha amino and alpha carboxyl groups. Okay, because they are in the peptide bonds. So the pi of the peptide is determined experimentally. Okay, we can determine, but then we have to do proper experiments to determine that. Simply by titration we cannot be able to determine the pi, just like we can do it for the amino acids. Okay, now pKa, remember, it is not fixed. It depends on the chemical environment around the group. Now you know that, because pK cannot be fixed here. You can see here in the case of glycine, the pK value is not fixed. It all depends whether we have more acidic environment or more basic environment, so it shifts. All right, now what are the important takeaways of this titration of the glycine? So let us see what are the important takeaways. First is that the curve gives pKa value of its two ionizable groups. So in case of glycine, we saw one pKa value is 2.34 here and another was at 9.60, which is here. Okay, now what does it mean? It means that there are two protons which were lost during the titration, right? Then glycine's carboxylic is more acidic than the normal condition or the normal acids. Now we compared it with the acetic acid. If we see the pKa value of the acetic acid, it comes to be 4.76. But the glycine pKa is less than 4.76. It is 2.34. That is, it is more acidic. All right. So lower the pKa, stronger the acid. Now the question is, why is it so that the glycine's carboxylic is more acidic than the acetic acid? Why? Because the nearby NH3 positive group, it pulls electrons towards itself, and this is known as electron withdrawing effect. Now when this effect happens, that makes the carboxylic lose proton more easily, and when it loses proton more easily, it lowers the pKa, and that's why the pKa of the glycine in carboxylic is stronger as compared to the acetic acid. Okay, now if we see in enzyme active sites, amino acids are placed very precisely because they have to make a very the center where the the substrate will bind to the enzyme. So their pK values are shifted, and they are shifted due to unique microenvironment. For example, there are nearby charges because of some other amino acids and hydrogen bonding or hydrophobicity, and that alters the proton affinity. Okay, so this helps to speed up the reaction and control the proton transfer, and that's why the amino acids and the enzyme active sites, they are placed very precisely. And if you do here and there, even a one position shift of the amino acid will completely can destroy the whole mechanism or the whole activity. Okay, the pK values of the carboxylic groups of the amino acids, they lie in a small range which is around 2.2, while the pK values of the amino groups are near 9.4. So now I am sharing you the pK values of all the amino acids. So I will just go through the slides, and at point of time you can just pause the screen and check the values there, and you will find the pK values which will be giving you an idea about how acidic or basic it can be. All right, so this is for the glycine. You can see here the pK for carboxylic, this is for amine, and this is for alanine, valine, leucine, isoleucine. You can see all the pK values, and this for carboxylic, they are within the range of 2.3, 2.9, and here it is 9.8, 9.7, 9.4. So they have a very limited range here. For methionine, proline, phenylalanine, tryptophan, and serine, you can see here the pK values for carboxylic and amine. They are again within that range what you saw here. And this is for threonine, asparagine, glutamine, tyrosine, cysteine, and you can see here the values here. They are again within the range which was defined there. And this is again the other set of lysine, arginine, histidine, aspartic acid, and glutamic acid, where you can see the pK values of both the amine and the carboxylic groups, and almost they are all coming in the range here. Now what I also want to pay I want you to pay attention here is that in case of tyrosine, cysteine, you will see the pKr values of the side chains. So that means their side chains are also ionizable. And in here also, in case of lysine, arginine, histidine, aspartic acid, and glutamic acid, you can see the side chains are also ionizable. All right, while such ionizable side chains are absent in other amino acids. So if we see and if we compare, so we have 2 here and 5 here, so total 7 amino acids are the amino acids which have ionizable R chains or R residues here when they come into the polypeptide configuration. All right, so I think you must have got some fair idea about the titration curve, how do we do that, and with the titration of the glycine, how we can do it with other amino acids. So everything is clear. In case if you don't, then you can just go through this video again, and or you can just write me over email. My email is given here. And now I will see you in your next class. Till then, take care and bye bye.